Bismillahirrahmanirrahim. Today I am going to take up cholesterol synthesis, its steps and the enzymes, along with the clinical correlations. So, in the waiting room, there is an obese man who is on statins and he is sipping grapefruit juice, and the doctor advises him to avoid grapefruit juice, and you are required to dig out the biochemical cause for this advice. Next, there is an old granny with high blood cholesterol levels, and she is being treated with lovastatin, and you are required to find that how this drug is helping her at biochemical level and how it is affecting her LDL receptors. Then there is a patient who is prescribed a statin to reduce the risk of atherosclerosis, and you are required to appreciate any potential problems that he may face. Next, there is a middle-aged man with high cholesterol along with the deranged liver enzymes, and you are required to prescribe him a medicine along with the biochemical justification for the prescription. Then there is a man munching a bowl of whole wheat cereal with sprinkled with olive oil, and you are required to find that how it is going to affect his cholesterol levels. Next is a boy, and he is a diagnosed case of phytosterolemia, and you are required to prescribe him some medicine. Then there is a dead body in the mortuary, and it is a young female. Diagnosis is phytosterolemia, and cause of death is cardiac arrest, and you are required to find the biochemical cause for this for her cardiac problems. Then there is a middle-aged man on statin therapy, and he has presented with brown colored urine after exercise, and you are required to find the likely biochemical cause. There is a middle-aged man with high total cholesterol, and he is prescribed statin therapy, and you are required to coin any follow-up advice for this man. Okay. Then there is a scenario that a medical student, a diabetic medical student, consumes a double cheeseburger and loaded fries. After that, he develops abdominal distress, nausea, and he faints. He presents in the emergency, and labs reveal hyperglycemia and high triglyceridemia. His condition is improved after insulin administration, and you are required to find the biochemical cause. So, cholesterol synthesis, around 1 gram of cholesterol is synthesized in the liver, whereas 0.3 grams is obtained from the diet. So, now coming to the steps of the cholesterol synthesis. It consists of 3 stages. In the stage 1, 3 acetyl CoA molecules condense to form the HMG CoA, and HMG CoA is further converted to mevalonate in the presence of HMG CoA reductase. In the stage 2, there is phosphorylation and decarboxylation, resulting in the formation of an isopentenyldiphosphate, which is a 5-carbon compound. In this stage 3A, isopentenyldiphosphate and dimethyl allyl diphosphate condense to form farnesyl diphosphate, and in the stage 3B, farnesyl diphosphate, 2-farnesyl diphosphate condense to form squalene, which is a 30-carbon compound, and in the stage 4, 3 methyl groups are removed from the squalene and it is converted to 27 carbon cholesterol. So let's dissect the stages one by one. You can see that there are 2 molecules of acetyl CoA, and for clarity these are represented by 2 different colors so that you can appreciate their incorporation. So in the presence of thiolase, these are converted to acetoacetyl CoA. Next, in the presence of HMG CoA synthase and another molecule of acetyl CoA, these form HMG CoA, and HMG CoA or hydroxymethyl glutaryl CoA in the presence of HMG CoA reductase and NADPH is converted to mevalonate, which is a 6 carbon compound. So this completes the stage 1. In the stage 2, mevalonate, in the presence of mevalonate 5 phosphotransferase and ATP, it is converted to 5-phosphomevalonate. Next, again in the presence of kinase and ATP, it is converted into 5-diphosphomevalonate, which is then decarboxylated in the presence of, again in the presence of the third molecule of ATP to isopentenyldiphosphate. Now this is the activated intermediate which is going to form the cholesterol. It also is a precursor for coenzyme Q and dolicol. So isopentenyldiphosphate is isomerized to dimethyl allyl pyrophsphate. Next, there is stage 3. Dimethyl allyl diphosphate and isopentenyl diphosphate condense in the presence of prenyl transferase to form geranyl diphosphate, which is a 10 carbon compound. Again a molecule of isopentenyl diphosphate is incorporated into it and it forms farnesyl diphosphate. And here you can see that it is a 3 colored compound representing the 3 molecules of the acetyl CoA. The 2 farnesyl diphosphates combine in the presence of squalene synthase and NADPH to form squalene, which is a 30 carbon compound. Now in the stage 4, squalene, in the presence of squalene monooxygenase, it is converted to squalene epoxide, which in the presence of cyclase is cyclized into a 4-ringed structure, which after many steps is finally converted to cholesterol. Now there is a question that what is the energy cost of the synthesis of squalene from acetyl CoA in terms of the number of ATP per molecule of squalene synthesized. So we have seen that only the ATP is consumed during the conversion of mevalonate to isopentenyl diphosphate. In the whole pathway, these are the only steps when ATP is consumed, and a total of 3 molecules of ATP are consumed. First molecule when mevalonate is converted to 5-phosphomevalonate in the presence of phosphotransferase. Next molecule of ATP is consumed when in the presence of kinase mevalonate 5-phosphate is converted to mevalonate diphosphate, and mevalonate diphosphate is decarboxylated again in the presence of the third molecule of ATP. So 3 molecules of ATP are required to form 1 isopentenyl diphosphate. Now 3 isopentenyl diphosphates are required for the formation of farnesyl diphosphate. So it means that 3 plus 3 plus 3, it makes 9 ATP for the formation of 1 farnesyl diphosphate. Next, the 2 farnesyl diphosphates combine to form squalene. So it makes 9 plus 9, so the total ATP cost is 18 ATP. As you can see, mevalonate to 5 phosphomevalonate 1 ATP, then 5 diphosphomevalonate again 1 ATP, and finally the third molecule of ATP during the decarboxylation to isopentenyldiphosphate. Now here are a couple of MCQs. Cholesterol synthesis occurs in the cytosol. Maximum cholesterol synthesis occurs in the liver. In stage 1 of cholesterol synthesis, mevalonate is formed. In the stage 2 of cholesterol synthesis, isopentenyldiphosphate is formed. In the stage 3 of cholesterol synthesis, squalene is formed. And in the stage 4 of the cholesterol synthesis, cholesterol is formed. Geranyl pyrophosphate or diphosphate has 10 carbons. Farnesyl pyrophosphate has 15 carbons. Squalene has 30 carbons. Lanosterol has 30 carbons. Cholesterol has 27 carbons. Now we are going to revisit the waiting room. So, the obese man who was sipping the grapefruit juice. Now the grapefruit juice contains furocumarins. These are the chemicals which inhibit the cytochrome P450 CYP3A4. Now CYP3A4 cytochrome P450 enzyme, it converts the statin drugs into compounds which are readily excreted from the body. When the furanocumarins are going to inhibit this enzyme, the statins are going to build up in this man. So these are going to lower the cholesterol levels, but these are also going to enhance the side effects like muscle weakness and fatigability. So inhibition of this enzyme is going to cause muscle pain and weakness in this man, so he is being asked to avoid these compounds. So then there is the granny with high cholesterol levels, and how this drug is helping her at the biochemical level. At the biochemical level, it is going to inhibit the HMG CoA reductase. So HMG CoA reductase is inhibited. When this is inhibited, the pathway of cholesterol synthesis will cease to function. So, as the saying goes, ना नोमन तेल होगा ना राधा नाचेगी, तो ना HMG CoA reductase होगा और ना कोलेस्टेरॉल बनेगी. हाँ, and a comment about the LDL receptors. The LDL receptors will be upregulated because the intracellular cholesterol levels will fall, and this will lead to the upregulation of the LDL receptors, which will internalize the circulating LDL, and this will lead to the reduction in the levels of the circulating LDL. Okay. Then there is a patient who is prescribed a statin to reduce the risk of atherosclerosis, and you are asked that which potential problem he may face. So as we discussed that the isoprenoid units are also precursors to the coenzyme Q and the dolicol. So as you can see that coenzyme Q is an important component of the electron transport chain. So decreased synthesis of coenzyme Q is going to take toll on the energy production in this man. So that is the coenzyme Q, and secondly it is also a dolicol is synthesized from the isoprenoid units, so its synthesis will also be affected. Okay. Then there is a middle-aged man and his liver enzymes are deranged. So obviously you cannot prescribe in statins. An alternative therapy may be cholestyramine, which binds the cholesterol, which binds the bile salts which are made from the cholesterol, and it causes the excretion of the bile salts into the feces, and otherwise 90% of the around 90% of the bile salts are recycled in the enterohepatic circulation. So next question is that whole wheat cereals, plant oils, how these can affect the cholesterol level. So these contain the phytosterols which bind to the cholesterol, and what happens is that here you can see this is the intestinal lumen, and here you can see there are green colored phytosterols and orange colored cholesterol molecules in the emulsion droplet in the intestine. These are going to form micelle, and micelle is going to dock at the brush border of the intestine, and it is going to and the contents of the micelle are going to enter the enterocytes. It is a diffusion process. The micelle as a whole is not going to enter into the enterocyte, it is just going to dock, and the contents of the micelle are going to enter the enterocyte. Here for the entry of cholesterol into the enterocyte, Niemann-Pick protein 1 is used. So once these are inside the enterocyte, the phytosterols are going to bind the cholesterol, and there are transporters on the brush border of the intestine, ABC cassette proteins ABCG5 and ABCG8. These form a complex sertolin, and it removes those phytosterols along with the bound cholesterol back into the intestinal lumen. So the absorption is prevented. So now back to the question that these phytosterols are present in the whole grain cereals. These are also present in the plant oils. So this man is consuming a breakfast consisting of whole grain cereals and sprinkled with olive oil. So the phytosterols present in these cereals are going to bind the cholesterol, and these are going to cause efflux of the cholesterol back from the enterocyte back into the intestinal lumen. So here you can see that cholesterol entered the enterocyte. Now these are back into the intestinal lumen. Okay, now there is a boy with phytosterolemia, and you are required to prescribe him a medicine and justify the biochemical cause. So what you are going to prescribe him is ezetimibe. Ezetimibe blocks the entry of the cholesterol into the enterocytes by from these proteins, NPC1 gene, Niemann-Pick protein 1 gene, which synthesizes these proteins, blue colored, and these help in the diffusion of cholesterol from the micelles into the enterocytes. So ezetimibe is going to block this diffusion into the enterocyte. So the cholesterol as well as the phytosterol, these will not enter the enterocytes. So this is because this child with phytosterolemia, he has no problem with the synthesis of the cholesterol. So there is no point in giving him statins. The drug of choice for him is ezetimibe, which is going to block the entry of the dietary cholesterol inside the cells. Okay. Now there is this dead body. Cause of death is cardiac arrest, very young age for cardiac arrest, and diagnosis is phytosterolemia, and the biochemical basis is obviously that there was buildup of the phytosterols inside the enterocytes along with the cholesterol, and the spilling of the phytosterols and cholesterol into the blood, leading to the cardiac problems. Okay, now there is a middle-aged man, and he has presented with reddish brown color urine after exercise, and he was on statin therapy, and you are required to coin the likely biochemical cause. So the likely biochemical cause is that, as we have seen, that the statins block HMG CoA reductase, and when HMG CoA reductase is blocked, the isopentenyldiphosphate is not formed. Isopentenyldiphosphate is the activated prenoid unit, which is not only required for the synthesis of cholesterol but also for the coenzyme Q. Now, coenzyme Q is an important component of the electron transport chain, and electron transport chain generates energy from ATP. Now, when there is not enough synthesis of the coenzyme Q, or there is deficient synthesis of the coenzyme Q, the electron transport chain is going to bear the brunt of this deficiency. When he is exercising, muscles are required to produce ATP at an accelerated rate, and when this is not produced, muscle damage is going to occur, and the myoglobin is spilled into the urine, giving the reddish brown color. Okay, now there is a middle-aged man with high total cholesterol. He is prescribed statin therapy, and you are required to give him a follow-up advice. Since the cholesterol synthesis occurs mainly in the liver, so the follow-up advice would be liver function tests after 2 to 3 months of the statin therapy. Okay, now there is this medical student who is diabetic, and he has developed abdominal distress and nausea after eating a double cheeseburger and loaded fries, and labs have revealed hyperglycemia and hypertriglyceridemia, and his condition has improved after insulin administration, and you are required to give the biochemical cause. So the likely biochemical cause is that since he is diabetic, he is going to have insulin deficiency. Now, insulin induces the expression of lipoprotein lipase, and this is the enzyme that extracts lipids from the chylomicrons into the adipose tissue. So when the expression is decreased, there is going to be hypertriglyceridemia and obviously hyperglycemia because of the insulin lag, and that is why after administration of insulin his condition has improved. So this is the most common type of hyperlipidemia, type 5, and it is secondary to diabetes. So that is the summary, that the story starts with the 3 molecules of acetyl CoA, and glucose is the common cause of acetyl CoA, and cholesterol is also synthesized from the acetyl CoA derived from the oxidation of the long chain fatty acids. So HMG CoA is formed, then mevalonate is formed, then Mevalonate is converted to isopentenyldiphosphate. Isopentenyldiphosphate is converted to farnesyl diphosphate, and farnesyl diphosphates are condensed to form squalene and lanosterol, and lanosterol after removal of 3 methyl groups is finally converted to cholesterol.